Assalamu alaikum warahmatullahi wabarakatuh. So today we are going to discuss the topic of cytogenic inheritance. Rather, we should call it as cytogenic disorders. So cytogenic disorders are mainly based on numerical and structural disturbances of chromosomes. So numerical and structural disturbances of chromosome. The normal human somatic cell has 46 chromosomes. Okay, but if someone has 45 or 47 total number of chromosomes, this is a cytogenic disorder, mainly a numerical defect. So in numerical defect, the number can either be slightly increased, the total number of chromosomes, or the total number of chromosomes can slightly be decreased. Example, 47 chromosomes or 45 chromosomes. Remember, normal human cell has 46 chromosomes. And then we have also structural disturbances of chromosome. So example, if there are normal human somatic cell has 46 chromosomes. So if someone has normal set of chromosomes, that is 46 chromosomes, but one of his chromosome, some part of that chromosome is deleted, so that means there is structural defect, and this is also included in cytogenic disorders. So structural defects can be like deletion, translocation, that is exchange of material between two chromosomes, and some other structural defects, etc. Before we go into the details of cytogenic disorders, we need to understand the term and the concept of karyotype or karyotypic studies. So karyotype or karyotypic studies are basically used for cytogenetic studies, in which we can then identify whether there is a numerical or structural disturbance. The procedure is that we take a cell and observe it in the metaphase stage. Why in the metaphase stage? Because in metaphase stage all the chromosomes are condensed and it is easy to view them under the microscope. So when the cell who is in metaphase stage is taken, its chromosomes are condensed, and then we dye this chromosome with Giemsa stain. Okay, and after we stain the chromosomes, we arrange the chromosomes according to their length in the decreasing order. So the longest chromosome to the shortest chromosome in descending order. So, and we know the chromosome 1 is the longest out of all. And then after that we will take a photograph of our result which is obtained. So basically, if we define karyotyping, it means that it is a photographic representation of a stained metaphase spread. That means stained chromosomes during metaphase, in which the chromosomes are arranged in decreasing order of their length, that is from chromosome 1, which is the longest, to the chromosome 22. And how are these chromosomes arrested at metaphase? It is with the help of the drug called colchicine. So sorry, the drug is colchicine and not colchicine. It is colchicine, which helps in arresting the chromosomes at metaphase stage. This is because colchicine, when it is applied to the cell, the mitotic spindle will not work further and the chromosomes will be arrested at the metaphase stage. After karyotyping is done and the photograph is taken, we can see the chromosomes arranged in such a fashion, and each chromosome has a short arm and a long arm. The short arm is called as P and the long arm is called as Q, and in the middle we have the centromere. And every chromosome is stained in a very what we call specific pattern, which is different from each other. So every chromosome will have a unique pattern or a unique banding pattern. So here we can see this is chromosome number 1 and chromosome number 2. Okay, both the lengths of both the chromosome, as we can see, is different, and even the staining pattern we can see is so different. So particular chromosomes are recognized by the length and by their staining pattern. So every chromosome has a unique staining pattern. So as a result, if any part of a chromosome is deleted, it can be easily found out by comparing with the original pattern. For example, here we can see, if we take this as chromosome number 1, okay, and this was a normal pattern, but here we can see something is lost, and such type of karyotypic abnormality is called as deletion, which can be detected by karyotyping procedure. And the point to remember is that in somatic cell study, when we study a somatic cell, every chromosome should have a homologous partner. That is, they should be in pairs. For example, chromosome 1 will have a paternal allele and a maternal allele. Same goes for chromosome number 17, 22, etc. and so on. But sometimes, if the genetic material is abnormal, we can find it out by karyotypic studies. For example, sometime we may end up with a female whose chromosome number is like 47XXX. This means she has a numerical problem which is detected by karyotyping, and this is a numerical problem because she has an extra X chromosome. So this woman will have two bar bodies instead of one. Another example is a person who suffers from Down syndrome. So basically in Down syndrome what happened, there is trisomy of chromosome number 21. So there should be one paternal and one maternal copy only, right? But this person has three, so maternal, paternal and extra 21 chromosome, due to which he suffers from Down syndrome. Now we will discuss the special language which is discussed or which is communicated basically by cytogeneticists. So this language is basically used by cytogeneticists. Here we are going to discuss an example of it. So the cytogeneticist, he writes genetic information in this form. So in this first thing what we write is, first we write the total number of chromosomes or the numerical value of total chromosomes in the organism. So 46 chromosomes, normal. Then we are going to write his gender, whether it's male or female. So if it's male, XY, if it's female, XX. And then we are going to write the type of structural defect, if there is any structural defect. So here in our case it's del, that means deletion. And then we are going to specify on which number of chromosome or the particular chromosome we are referring to. So here we have written as 3, so third chromosome. Then we are going to refer to which part of the chromosome, whether it is on the short arm or long arm. So if it's short arm, it's going to be P. If it's long arm, it's going to be Q. Then we are going to talk about the region number, that is here it is 5, and then we are going to talk about the band number. So here we have our chromosome number 3, okay, and then we were told that in chromosome number 3 the defect was in the P arm, so short arm, P in the P arm on the 5th region. Okay, here remember region, 5th region. So the number of region is from the centromere towards the telomere. That's how we count the region number. So this is region number 1, 2, 3, 4 and 5. There we have to see the region number 5. Then band number. The band number is also same from the what we call centromeres towards the telomere. So towards the closest to the centromere is this. So this is band number 1, band number 2, band number 3, band number 4. So here they are saying that in the chromosome number 3, okay, this is our chromosome number 3, in the P part, that is a short arm, in band number 5, region number 5, in region number 5, band number 1, here this is band number 1 of region number 5, there has been a deletion. So this is how you read the genetic information in the special language which is produced or used by the cytogeneticists. Now we are going to see another example. So here it is written as 47 triple X del 6Q34. Before we discuss this example, we need to understand the concept of euploidy, aneuploidy and polyploidy. So in this example, we can see there is a numerical problem as well as a structural problem. But before that, let us understand the concept of polyploidy, aneuploidy and euploid. So euploidy or euploid, which means true, U means true. The definition is that chromosomes present in a cell are direct multiple of 3. For example, in normal human cell, we have 1 and that is 23 pair of chromosomes. Okay, so 23 in the gamete, 23 chromosomes. And diploid is 2n. So, and triploid is 3n, 69, and tetraploid is 4n, 92. So all these are direct multiples of 23. So that is why they are going to be called as euploid or euploidy. 3n and 4n, triploid and tetraploid are called as also polyploid, and this is seen in cancerous cells. Diploid is the normal somatic cell and haploid are the gametes. And aneuploidy is a numerical problem in which the number of chromosomes are not direct multiples of 23. So the examples are going to be 47 triple X. This means it is a female with 2 bar bodies. 47 XYY. In this is a male with an extra Y chromosome. And other examples are 47 double XY. This is Klinefelter syndrome. And also 45XO. This is an example of Turner syndrome where the female has just one X chromosome. So one X is missing in her. Instead of XX, it is just X. So this female in every cell will have 45 chromosomes instead of 46 chromosomes. So when we talk about numerical problems, we should first discuss if the cell is aneuploid or euploid. Then if the cell is, for example, euploid, then we should discuss if it is haploid, diploid, triploid, or tetraploid. So now if we come back to this example, here we can see this person has 46 chromosome, and 46 is not a direct multiple of 23, so he has aneuploidy, and the cause of aneuploidy is one extra X chromosome, and this is going to be a female who has two bar bodies. And then the type of defect or structural defect is deletion, and the deletion takes place in the sixth chromosome in the Q arm, so in the long arm, in the third region from the centromere, and in band number 4 of region number 3. So this is how we deciphered the genetic information given to us by the cytogenicist. Now we will see an example where there is translocation taken place. So this is the genetic information provided to us. So here we can understand that this person's total number of chromosome is 46. So that means he is euploid, and in euploid he is diploid. Why? He has 46 chromosomes. So as we remember, 46 is diploid, 69 is triploid, 92 is tetraploid, and haploid is 23. So that is why this individual is euploid but diploid. In euploid, that is total number of chromosomes 46, and then his what we call genetic sex or his gender is male because he has XY. Then the structural defect that is present in him is translocation, because T is written, so it is translocation, and the translocation takes place between chromosome number 2 and chromosome number 5. This means that some genetic material has been exchanged with has been exchanged within the chromosome number 2 and the chromosome number 5, and then it's written P13 is to Q33. This information tells us that the short arm, that is P arm of chromosome number 2, has exchanged its material with long arm of chromosome number 5, and the chromosome number 2 short arm region number 1 band number 3 has been exchanged with the chromosome number 5 long arm region number 3 band number 3. So this is how you decipher such genetic information, and this is an example of translocational defect. This kind of language is used in genetic disorders of cytogenetic inheritance, and it is mainly deciphered with the help of karyotypic studies. So now, as a quick recap, cytogenetic studies, we use karyotyping and look for numerical and structural abnormalities of the chromosomes. So the technique of karyotyping is that the cells are arrested at the metaphase stage with the help of colchicine drug, and then they are stained with Giemsa stain and arranged in decreasing length, and then the photo is taken, and then we can study the chromosome numerically, as well as different regions of chromosome can be studied for any structural defects. Then we look for numerical information, and numerically the number of chromosomes the cells can have euploid or aneuploid. Euploid means direct multiple of 23, and if it's euploid direct multiple of 23, then we check whether it is 1n, that is 23, 2n, that is 46, 3n, that is 69, and 4n, that is 92 chromosomes. Or if it is not direct multiple of 23, the total number of chromosomes not direct number of 23, then it is called as aneuploidy or aneuploid. So if a cell is aneuploid, it does not have total number of chromosomes direct a multiple of 23. For germ cells, it will be written as n minus 1 if it is 22 number of chromosome, and n plus 1 if it is 24 number of chromosomes. So here we can see they are not the multiples of 23. Whereas for somatic cells, we know normal somatic cells has 2n, and normal germ cell has n is equal to 23. So that is why n minus 1, n plus 1 is an example aneuploidy for germ cells, whereas somatic cells the normal number is 2n of chromosomes, but if it is aneuploid it will be 2n plus 1, that is total 47 chromosomes, and 2n minus 1 equals to 45 chromosomes. To what we call for structural defects, we need to remember that P is the short arm, Q is the long arm, and so P is the short arm, Q is the long arm, and we divide the arms into regions and band, and the regions and band start from the centromere towards the telomere. So this is going to be region 1, 2, 3, 4, 5, and in region 5, for example, this is band 1, band 2, band 3, band 4. So now we should understand how such numerical abnormalities or structural abnormalities occur. But to understand that, first we need to understand how the normal meiosis works. Why we need to know meiosis? Because many numerical problems or disturbance are produced before the zygote formation and even after zygote formation. So example, one of the numerical problem is going to be aneuploidy, where the number of chromosome is not a direct multiple of 23, and this can happen before fertilization or after fertilization, which is also called as pre-zygotic or post-zygotic. So to understand this, we should first revise meiosis. Now we are going to discuss meiosis in a bit detail, but before we start discussing meiosis, we are going to revise some important concepts. So in this diagram here, we can see homologous chromosomes. And why are they homologous? Because this is also chromosome number 1 and this is also chromosome number 1. This is maternal chromosome number 1 and this is paternal chromosome number 1. Here we have two chromosomes, okay? And both the chromosomes are monovalent, okay, because they don't have the sister chromatids with them. And here the total genetic material, we will assume it as 2n, because this represent 23 pairs of maternal chromosomes and this represent 23 pairs of paternal chromosomes. So 2n, 2 into 23 is 46 pair of chromosomes are present in this cell, okay. For example, now we are just depicting one pair of homologous chromosomes and they are monovalent. In this example here also we have again the chromosome number 1 and chromosome number 1, maternal chromosome number 1, paternal chromosome number 1, so they are homologous chromosomes. Here also this full thing will be counted as one chromosome and this will be counted as the second chromosome. So here also we have total two chromosomes and both the chromosomes are divalent. So here they were monovalent, but here they are divalent. Why? Because they have the sister chromatids with them. And here also the genetic material is 2 and that is 2 and 23. So total 46 chromosomes are present in this cell. So as I mentioned before, in a divalent chromosome, so this is just one chromosome. In this divalent chromosome, these are called the sister chromatids of the chromosome, and this is going to have 1n genetic material, 23 pair of chromosomes. And this is an example of a cell which has 1n chromosome, that is only 23 pair of chromosomes, and out of this, this is one chromosome. This is just one chromosome and it is monovalent. So now we are going to discuss the meiosis. So for meiosis, let us assume that this is a cell which is going to make the gametes and its genetic material is consisting of 2 and that it has 23 pairs of maternal chromosome as well as 23 pairs of paternal chromosome. It is difficult to illustrate all the 23 pairs, so just we will depict or illustrate just one pair of chromosomes out of all. And for example, here we are going to take the third chromosome and these are homologous chromosomes and they are present in monovalent situation here. So the first thing is that the cell will double its chromosomes or do replication. So now we will again have, we can see here they have two chromosomes but both of them in divalent situation. And then the next stage is synapsis takes place or crossing over, or this is also called as chiasma, where there is exchange of genetic material between the maternal and paternal chromosomes. Because of this, after this we are going to get genetic diversity. And after synapsis, the homologous chromosomes will separate from each other and they are, each chromosome is now going to have some paternal and maternal gene mix. Okay, as you can see, as here they were pure, they were only pink and blue, but now they are pink blue mix among themselves. So this means each chromosome is going to have some paternal characters and some maternal characters and they have exchanged their what we call genes among each other after synapsis. So all the steps till here, till what we call, we get synapses, and after synapses we get the divalent chromosomes with what we call mixed genes. This is till here we have this is called the first stage of meiosis. After this we have the second stage of meiosis. So after the first stage of meiosis ends, the second stage of meiosis starts by the splitting of these into two cells. Okay, so 23 pairs of chromosome are going to go to this cell and the rest 23 pairs of chromosome are going to this this cell. So now this is not going, this was 2n but now this will be just 1n. It is a single chromosome but it is divalent. The same happens here, this is a single chromosome divalent. After that there is disjunction between the sister chromatids. So one chromatid goes to each cell and these form the gametes. So now in the gametes we will have one chromosome and that chromosome is monovalent. Okay, and these monovalent single chromosomes have paternal as well as maternal gene mixture, all thanks to our synapses during the first meiosis stage. And the second meiotic stage was just including the separation of the divalent chromosome and then formation of single divalent chromosomes, and then eventually their dysfunctions. This was meiosis stage 2. Now, important thing to remember is that defect during stage 1 of meiosis or defect during stage 2 of meiosis may result in aneuploidy. Now we are going to see how aneuploidy occurs. Another important thing to remember is that these are gametes, okay? So when this gamete is going to fuse with, if for example this is a paternal gamete, and when it is going to fuse, for example, with a maternal gamete, 1n plus 1n will form 2n. So the zygote will always have 2n diploid stage. So 1n haploid germ cells will fuse with each other and form 2n zygote, which is a diploid. That is 2n is 2 into 23 is 46 chromosomes. So now we remember that aneuploidy is a numerical problem in which the total number of chromosomes is not a multiple of 3. So aneuploidy can occur what we call problem during meiosis and post-zygotic. Okay, so now we are going to discuss the problem during meiosis. So aneuploidy can occur during meiosis, either in during meiosis stage 1 or in meiosis stage 2. So in meiosis stage 1, as we remember, the main events were synapses and then disjunction. So what happens sometimes if this synapses and crossing over takes place of the maternal and paternal homologous chromosomes, for example chromosome number 3 we have considered here. After the chiasma, this is, we remember it's called a chiasma. So after the chiasma is formed, there is a mistake and there is not proper disjunction of the chromosomes. Okay, so they don't separate properly. So what happens, for example, 3 of the chromosomes out of 4 will go in one cell, and one chromosome, which was here we had to 2 right in each side, one will come here. After that, we remember stage 1 of meiosis comes, stage 2 starts. So in stage 2 what happened, the cells are going to separate, these two are going to go here, these two are going to here. Now what happens here is that from here 2 will go here and 1 will come here. Okay, and then this one chromosome is going to come here, whereas this one chromosome is not going to be present in this case. So this means that in this cells all the chromosomes are going to be present except the chromosome number 3. So that is why the total number of chromosomes in this will be 1n minus 1. In this chromosome we have 1n chromosome, so we have the number 3 chromosome as well as all other chromosome, pair of chromosome, it's normal. Here also we have 1 chromosome with all the other chromosome, chromosome number 3 with all the other chromosomes, so this is normal. So if these two gametes are going to what we call result in the formation of the zygote, then it will be normal. But here in this case you can see here we have an extra copy of chromosome number 3, okay, in this, and here we have no copy of chromosome 3 but other pair of chromosomes are present. So here the configuration is going to be 1n plus 1, whereas here the configuration is going to be 1n minus 1. So if this 1n minus 1 is going to fuse, for example if this gamete is a sperm, if this is going to fuse with a healthy ovum 1n, so it will be 1n minus 1 plus 1n. So the total is going to be 2n minus 1. So this means that in such a zygote there will be monosomy of chromosome number 3, that is that it won't have 2 chromosome number 3, it will have just 1 chromosome number 3, and the total number of chromosome, that is 2n minus 1, will be 45, which is aneuploidy because it is not a direct multiple of 23. Then in this case here if we see, here we had an extra chromosome number 3, here we had one less, that is chromosome number 3 is missing, and here we had extra chromosome number 3. So if again we consider this as a gamete and this is a sperm, and if it fuses with the normal ovum, 1n plus 1 plus 1n is going to form 2n plus 1, and the total number of chromosome is going to be 47. That means there is trisomy of chromosome number 3, that is the zygote will have an extra chromosome number 3, and the total number of chromosomes will be 47, which is again not a direct multiple of 23, and it results in aneuploidy. Another way by which aneuploidy can occur during pre-zygote formation is if it doesn't happen during meiosis stage 1 is absolutely normal, but there is a problem in meiosis stage 2. So what is the problem? That meiosis stage 1 everything went good, and after meiosis stage 2 started, the cells got separated what we call normally, and each had a single chromosome with divalent. But now these two sister chromatids have to what we call separate from each other and go respectively in single gametes as what we call 1n, 1 chromosome, and monovalent. But what happens sometimes, there is a problem in disjunction or separation here. So for example, if there is a problem in separation, one of the gamete is going to get a chromosome which is going to be divalent containing both the sister chromatids, whereas this gamete is going to get no chromosome number 3. So as we know, we were considering these to be chromosome number 3, right? So this gamete is going to have one extra chromosome number 3, whereas this won't have a chromosome number 3. So the total configuration be 1n plus 1, whereas the configuration of this will be 1n minus 1. So this 1n plus 1, if it, for example, these will consider as sperm cells, if they are going to fuse with a healthy ovum, so 1n plus 1 plus 1n is going to form 2n plus 1, that is equal to 47 chromosomes. So this means there is going to be trisomy of chromosome number 3, and that is also 47 chromosome is another type of aneuploidy because it is not the direct multiple of 23. Whereas the gamete which didn't get the copy of chromosome number 3, its configuration is 1n minus 1. So this means that 1n minus 1, that is sperm cell plus a healthy ovum, 1n is going to form 2n minus 1, that is total number of chromosome is going to be 45 chromosome, which is again aneuploidy because it is not a direct multiple of 23, and this zygote will have only one chromosome number 3, so it will suffer from monosomy of chromosome number 3. So what we understood that how aneuploidy can be produced before zygote formation during meiosis is that either there will be non-disjunction problem in meiosis stage 1, or either there will be non-disjunction problem in meiosis stage 2. So basically there's a problem in which the chromosomes don't separate properly either in stage 1 of meiosis or in stage 2 of meiosis. Now we are going to discuss aneuploidy from another angle. For example, this is maternal ovum and this is paternal sperm. So we have here a homologous pair of chromosomes A, B and CD. So in meiosis stage 1, we know that there is replication of the chromosome going to take place, and we will have two chromosomes, but they are going to become divalent. Okay, so till here we had what we call our stage 1 of meiosis, and don't forget that when they become divalent, after they'll form chiasma, and there will be exchange of genetic material. So none of the chromosome is going to remain pure, but they will have both maternal and paternal genes. So till here we had the stage 1. Now in stage 2 of the meiosis, we remember that there is going to be what we call splitting of these. One pair of chromosome will go in one cell, other pair of chromosome will go in one chromosome is going to go here, one chromosome is going to go here. Same happens in the paternal side, and after that there is going to be disjunction here. So we will have a single gamete containing of a single pair of a single set of chromosomes, that is 1n, that it has 23 chromosomes, and they all are monovalent, they don't have the sister chromatid. So till here we have the this is our meiosis stage 2. So now the possibility we come up with that is the mother is going to provide only A or B, what we call gametes, and father is going to provide only C or D gametes. So when such gametes fuse, we have the following possibilities: AC, AD, BC and BD. So this is our zygotes for possible genetic makeup, and they all are diploid, that is they contain 46 pair of chromosome and divalent. So sorry, they are going to have diploid, what we call chromosome, that is 2n, 46 pair of chromosomes in the zygote, and they are not diploid, but they are not divalent, but they are diploid. Now if the same couple produces a child zygote whose genetic makeup is ACD, so this means there was a defect in first stage of meiosis, because we remember that C and D were together, so maybe there was non-disjunction which took place over here, and due to that result we had A which is coming from the mother but CD which is coming from father, and that is the defect in stage 1 meiosis paternal. Then if there's a possibility of the zygote genetic makeup of A, B, C, so we again know that the A came from mother and B also came from mother, but the C came from father. So this means there was a disjunction problem during the stage 1 of meiosis, but maternal stage 1 of meiosis. So here we remember the mother had A and B gametes possibility, and if there is A, B, C genetic makeup of the kid, that means there was a problem with the segregation or non-disjunction during stage 1 of meiosis in mother. Then if we have a child with the possibility of genetic makeup AAC, so this means there was a problem in disjunction during second stage of meiosis, where the cystic chromatid failed to separate from each other in the mother. Why? Because the mother had A and B, and so most probably AA came from mother. So there was disjunction problem here during the stage 2 of meiosis. So AA didn't separate as A and A, but they came together as AA, and that AA combined with the C formed double AC genotype that we saw here. Then if we have a possibility of B double C genetic makeup, this means this is the failure of disjunction during the meiosis second stage, and that is in father, because we know in the dad we had C and D gametes. So this means that there was a problem in disjunction stage 2, that this double C did not, the sister chromatids did not separate and form a single what we call gametes, but they remain together in a one gamete, and that result in the formation of genotype of B double C. There can be another cause of aneuploidy during the second meiotic division, and that is called as anaphase lag. So we know during normal second stage of meiosis, the what we call the single chromosome which are divalent have to undergo disjunction and form two chromosomes which will each have one sister chromatid and will be monovalent, but will have one full pair 23 pair one set of 23 chromosomes. Now sometimes what happen is that when this what we call chromosome is going to divide its material into two different cells, out of the 23 set of chromosomes which came here, the nuclear envelope is going to develop. 22 chromosomes move within the area where the nuclear membrane is going to be formed, but one chromosome out of the 23 moves very slowly, and by the time the nuclear membrane is formed, it does not reach there, and the nuclear membrane is formed and it remains outside in the cytoplasm and gets degenerated. As a result, this gamete is going to have 1n number of chromosome, but this gamete is going to have 1n minus 1 chromosome because it lacks that one chromosome, and this is called as anaphase lag. So this is what we are trying to explain, that before the last phase of anaphase of the meiosis, all chromosomes should be within the nuclear envelope, okay, but sometimes it happens during segregation, one chromosome is moving too slowly and it can't enter the nuclear envelope and it remains outside. So 22 chromosomes enter the nuclear envelope, but one remains outside, and that one which remains outside gets degenerated. As a result, the final product will have one chromosome less than the normal, and that is 1n minus 1, and this will be the cause of aneuploidy. Now let us recap the cytogenic disorders. So we know cytogenic disorders are of two types: numerical disorders and structural disorders. Numerical disorders are further divided in two types: euploidy and aneuploidy. Euploidy is the case where the total number of chromosomes is direct multiple of 23. So the possibility is 1n, which is 23 chromosomes, which is also called as haploid, and it is seen in germ cells. 2n, which is 46 chromosomes, it is called as diploid, and it is seen in somatic cells. 3n is 69 chromosomes, and 4n is 92 chromosomes, and 3n and 4n together are called as polyploidy, and 3n and 4n is usually seen in cancer cells, or if there is a fetus which has 3n or 4n, it is usually aborted and it dies even before it is born. Then we move on to aneuploidy. So aneuploidy is a numerical disorder in which the total number of chromosome is not the direct multiple of 23. So this can be caused due to two reasons: aneuploidy before the zygote is formed, or aneuploidy after the zygote is formed. So aneuploidy which is formed which is due to before the formation of zygote is called as pre-zygotic or pre-fertilization, and aneuploidy which is after the zygote is formed is called as post-zygotic or post-fertilization. So pre-zygote or pre-fertilization aneuploidy, the cause is either maternal germ cell defect or paternal germ cell defect. So if there's a defect in maternal germ cell, that means there is defect in oogenesis, and that can be due to non-disjunction during the first meiotic stage, or non-disjunction during the second meiotic stage, or anaphase lag. Same goes for paternal germ cell defect, that is the defect in spermatogenesis. So that can be caused due to non-disjunction during first stage of meiosis, non-disjunction during second stage of meiosis, or anaphase lag. So the important thing to remember over here in aneuploidy, that is pre-zygotic aneuploidy, the result, that is either defect in the maternal or paternal gametes, is eventually going to result in 1n plus 1 possibility or 1n minus 1 possibility. So for example, now if we take the 1n plus 1 possibility of the gamete and fuse it with the normal gamete, we are going to result with 2n plus 1. So there is the answer is it will result in trisomy of the particular chromosome. Whereas if we take the possibility of 1n minus 1 and fuse it with the normal gamete, it will result in 2n minus 1, that is going to be monosomy of the particular gamete. So prezygotic or pre-fertilization aneuploidy can result either in monosomy or trisomy of the particular chromosome. The other cause of aneuploidy is post-zygotic or post-fertilization. So here what happens is, for example, if we take 23 chromosomes from the father and from the mother, and when the zygote is formed, we get 2n, that is 46 pair of chromosomes, out of which 2 are the X chromosomes. Okay, so the zygote is a girl, female zygote. So now when mitosis takes place and it divides into two cells, each cell is going to have 46 chromosomes. Okay, then further mitosis is going to take place, and the first line of cells is going to form further two lines of cells that is going to have 46 chromosome with 2x and 2x normal. Whereas in the second line of cells, something happened wrong during mitosis, and one line of the cell is going to have 45 chromosomes, other line is going to have 47 chromosomes. So the one line is going to have an extra X chromosome and one is going to have a one less chromosome. So this line is going to suffer from monosomy and this line is going to suffer from trisomy. So this means that in the same person, different cells have different karyotypy. That is, some cells are normal, that is they will have 46xx, while some cells will have 45xo, while other cells will have 47 triple x. Okay, so there is monosomy in some cells, there is trisomy in some cells, and there are normal, some cells are normal. In such cases, okay, results in this kind of situation where we can see monosomy, trisomy and normal cells together, this is called as mosaic. So this is an example of post-zygotic or post-fertilization problem. So now when we talk about aneuploidy, we have to basically remember that aneuploidy, especially prezygotic, can be caused due to 1n plus 1 scenario or 1n minus 1 scenario. So when a normal gamete will fuse with such 1n plus 1 gamete, it will form 2n plus 1, and that will result in trisomy. And then when a 1n normal gamete is going to fuse with 1n minus 1 gamete, it's going to form 2n minus 1 zygote, which is monosomy. So trisomy and monosomy can be further divided into: trisomy can be autosomal trisomy, that is there is an extra chromosome, that is from 1 to 22, which are autosomes, there can be any extra chromosome, or there can be sex chromosomes trisomy, that is there can be either extra X or extra Y chromosome. Same goes for monosomy. In monosomy we have autosomal monosomy and sex chromosome monosomy. So this means that there is one less chromosome or one less autosome, that is 1 to 22 chromosome, something is missing, and sex chromosome monosomy means either X or Y chromosome is missing. So we have to remember some basic principles, such as that extra dose of chromosome is tolerated better than the underdose of chromosome. So this means that trisomy of autosomal or sex chromosome is more compatible with life as compared to monosomy of autosomal or sex chromosomes. And also autosomal aneuploidy are more dangerous than sex chromosome aneuploidy. So this means that autosomal trisomy or autosomal monosomy are much more dangerous than sex chromosome trisomy or sex chromosome monosomy. Now we are going to look into some examples of trisomy and monosomy of autosomal and sex chromosome. So first we start with sex chromosome monosomy. So there is only one case of sex chromosome monosomy which is compatible with life, and that is XO, where there is one X missing, and this is called as Turner syndrome. So such female will have no bar body, and this is the only case of sex chromosome monosomy which is compatible with life. Then we move on to autosomal monosomy, in which there will be one less chromosome, then that chromosome is an autosome, and unfortunately none of this is compatible with life, and almost all the fetuses will be aborted and die even before they are born. So autosomal monosomy, there is no example because it is very dangerous. Then we move on to trisomies. So first we are going to discuss trisomy of sex chromosomes. So this means there is an extra X or extra Y chromosome. So if there is a female with triple X, that means she has an extra X chromosome, and that extra X can either come from the father or from the mother. Then we have another example, that is XXY. So since there is Y, this is a male, and this is called as Klinefelter syndrome. So same goes here. The extra X can either come from the father or the mother. Then the third example of sex chromosome trisomy is XYY. So here we have an extra Y chromosome, and this Y chromosome is surely going to come from the father, because only the father can give the Y chromosome. Now the point to remember, in the first example triple X2, we have two bar bodies, and here in Klinefelter syndrome, since it's a male, only one X is going to function and one X is going to be the bar body. Now here we have two Y's, okay? So there we can't call it bar body. When we have an extra Y, we call it as Y body. So this male has an extra Y body, Y, and that is called as Y body. Such males are stronger, taller, and more aggressive. And in the jails, most of the people who end up in jails are statistically found out to have such genetic makeup of XYY. Then we are going to discuss now autosomal trisomy. So there are three cases of autosomal trisomy which are compatible with life. That is, autosomy of chromosome number 13 is called as Patau syndrome. Autosomy trisomy of chromosome number 18 is called as Edwards syndrome, and trisomy of chromosome number 21 is called as Down syndrome. The way to remember is this: at 13 years of age we hit puberty, so that is Patau syndrome. At 18 years of age we can vote in elections, so E for elections, E for Edwards syndrome. And at 21 years of age we are legal to drink alcohol, and we go down with alcohol, that is why we can remember as Down syndrome. We're going to discuss about the first sex chromosome monosomy, that is, and the only compatible with life sex chromosome monosomy, that is Turner syndrome. So in Turner syndrome, this is a lady, okay, so it is a woman. This is a lady who has turned down an X chromosome. So this means that she does not have two X chromosome, but she has one X chromosome. The absence of X chromosome may be complete absence of that particular X chromosome or a partial absence of the X chromosome. So therefore, Turner syndrome is of two presentations: classical case of Turner and non-classical case of Turner. So classical case of Turner is that case of Turner in which the woman has only one X chromosome and the other X chromosome is completely absent. Whereas non-classical case of Turner is that case of Turner in which the second X chromosome is present but it is defective. Okay, so there is partial absence in the non-classical case of Turner. So let's discuss the classical case of Turner. So classical case of Turner, the it's going to be total number of chromosomes 45, so it is 45X. So because one X is missing and there is complete absence of one X chromosome, this absence of X chromosomes can be due to paternal or maternal side problem, that is problem in either oogenesis or spermatogenesis. And we should know that normal female requires two X chromosomes for healthy life. Now coming back to non-classical case of Turner. So non-classical case of Turner is that case of Turner in which the second X chromosome is present but it is defective. Okay, so something is missing. So now let us see at the examples. So we have 46XXP minus 46XXQ minus 46XI bracket XQ, 46XI bracket XP, and 46XRX. So what are these? So now if we start with 46XXP minus, this means here in the second X chromosome the short arm is missing. 46XXQ minus means here in the second X chromosome, long arm of the second X chromosome, that is Q, is missing. Then we have another example, that is 46XIXQ, okay, or 46XIXP. So we know that this is a normal chromosome which has a short arm and a long arm, so XP, XQ, okay, because X chromosome, then when it has to what we call divide before that, and when it goes meiosis, it goes under replication, so we will have X chromosome with 2P and 2 Qs, okay, so it will have 2 short arms and 2 Q arms. So normally when they are going to have non-disjunction, they are going to vertically, so they are going to divide like this and separate, okay. So this is the what we call normal way of division. So then we will get the two sister chromatids will separate and we'll get two chromosomes. So this means that when normal separation or non-disjunction happens, it happens in the vertical direction. Now, but sometimes what happens is that instead of vertical what we call dysfunction, very rarely in gametogenesis there is horizontal dysfunction, okay. So this means that two P arms will separate together and two Q arms will separate together. So in this what happens is the arms which will have this more part of centromere with them will survive, whereas the ones without centromere will get digested. So it can either be for the short arm, that is it will be XPP that will take the centromere, or it can happen for the Q arm. Okay, it can be XQQ. So here the P arms will be digested, and here XQ arms will be digested. So such chromosome which contain only the P arms or only the Q arms are called as isochromosomes. Or such chromosomes in which both arms have same genetic information are also called as isochromosomes. So then this will be written as, if for example we have only the short arms left, as 46X, because one X is normal, the other X has the problem, so it will be isochromosome XP because she has only the P arms. Or if only the Q arms survive, then we write as 46XI, which stands for isochromosome, and XQ. A point to remember here is that even though P arm is short arm of the X chromosome, most of the genes for female development are present on the P arm. So such female who has 46XIXQ or 46XIXP, she has 46 chromosomes, and she has one normal X chromosome, but the second partial chromosome she has. So she will develop some partial features of, or some features of Turner syndrome. So now there can be another problem which can occur. Sometimes what happens, there is, for example, this is a normal X chromosome. So there is terminal deletion which occurs from both the sides of the X chromosome. What happens next is these two sides will stick with each other and they will form a shape like this, which looks like a ring, and so it will be called as ring chromosome. These ring chromosomes are either lost because they digested away, because a weird shape they have, because they are either lost, or what we call, they don't behave properly during meiosis and meiosis. And even if they are structurally present, okay, even if this rings, what we call structure, is present, they will never express their genes in normal fashion, so they are functionally defective. So what does this mean? That these ring chromosomes are either lost because they cannot undergo mitosis or meiosis like how normal chromosomes will undergo, or even if they're structurally present, they won't express any of their genes because they are functionally defective. So in such a case, such a female who has such second X chromosome, her karyotype is going to be 46 chromosome total, X a ring case of other X. Okay, so X R X. So this is what we call the another possibility of non-classical Turner syndrome. So now we should remember that all Turner syndrome patients are not 45XO. Okay, only 50%, half the cases are classical situation of Turner, that is they have 45XO karyotype. The remaining 50% can be 46XXP minus, where the second X doesn't have the P arm, 46XXQ minus, where the second X chromosome doesn't have the Q arm, 46XIXQ, that means it is what we call this isochromosome of XQ, or isochromosome of XP, that is 46XIXP, and the last one is 46XRX, where the second X chromosome is a ring chromosome. Now we will see the general features of a person who suffers from Turner syndrome. So one of the most important things about Turner syndrome patients are they are short-heighted. They have a short height. So for example, here I drew a patient who has a short height. These patients are going to have low posterior hairline, webbing of neck. So webbing of neck is because there is abnormal development of lymphatic around lymphatic vessels around the neck, so the neck does not develop properly. So webbing basically means wide neck. Okay, this is because abnormal development of lymphatics around the neck. Then we have carrying angle is pathologically increased. So carrying angle is the angle between the arms and the body. So here we can see the angle between the body and the arms is called as carrying angle. Normally females are obese in the hip region, so their arms are a bit away from the body. But cubitus valgus, okay, it is a condition in which there is increased in pathological increase in carrying angle, and that is seen in Turner syndrome patients. cubitus valgus. Then they have broad space nipples, that is there is big distance between the nipples. Then they can have cardiac problems and coarctation of aorta. This means the heart does not develop properly, and there can also be coarctation of aorta. Then they can have problem with the development of kidneys, so that is also depicted here. And they can have problem with the development of external and internal genitalia, especially the gonads. The ovaries, they will not develop properly. The external genitalia will also not develop properly. The uterus does not develop like females, normal females. Okay. And then one more thing, there will be lymph edema not only in the neck here, but also in the legs. Okay, that can also be noticed. So here we can see in this diagram, in this patient of Turner syndrome, she will have a low posterior hairline. So behind her head, the posterior hairline is going to be low and it's going to end very soon. And then she is going to have broad space nipples, webbing of neck, heart problems and coarctation of aorta, cubitus valgus, big carrying angle is increased. Then the kidneys also don't develop properly, the gonads also don't develop properly. Then there is going to be lymph edema, and very important thing, primary amenorrhea, and eventually the patient is short heighted. So this is a Turner syndrome patient. Now a very important thing to remember is that every Turner patient won't have all the symptoms which we have explained and shown over here. Okay, but the two main symptoms which are present in almost all the Turner syndrome patient are short height and primary amenorrhea. So if a patient is short heighted and she has primary amenorrhea, then we suspect Turner syndrome and send such person for karyotype. This is because every Turner is short heighted and has primary amenorrhea. The other features are not going to be present in all patients. Some patients will have. So this basically means that every Turner won't have all the symptoms. So every Turner patient won't express all these symptoms which we discussed, but all the Turner will have two symptoms which are common, that is short height and primary amenorrhea. So when we suspect them, we will send them for karyotyping. And this is because why all Turners don't exhibit all symptoms, because we remember the concept of variable expressivity which we discussed in Mendelian inheritance, according to which it states that people with the same disease may express it with different severity, different manifestation, and at different onset of age. That is the concept of variable expressivity, which can be seen in even in Turner patients. Now the important thing here to understand is that the case of what we call 45XO and 46XXP minus 46XXQ minus 46XIXQ, 46XIXP and 46XRX chromosome, when we saw this classical case of Turner, non-classical case of Turner, these were mostly caused due to pre-zygotic problems. That is, there was either a problem with oogenesis or what we call spermatogenesis. Okay, that led to the formation of the fetus which was having defective karyotype. Now we are going to discuss an example in which the X chromosome was brought from both the paternal and maternal side in normal fashion, but the division of chromosomes was not proper during early stages of mitosis, during the early life of the fetus. Okay, and that resulted in the person suffering from Turner syndrome. Okay, so this is a post zygotic stage problem, and such Turner syndrome female is called as mosaic. So what happens here is that in such female, some cells of her are going to have only one X chromosome, so they will have 45XO karyotype, while some other cells of the same female are going to have 46XX, like normal karyotype. So such a female who has more than one karyotype in different cells is called as mosaic, and this problem is basically caused due to post-zygotic stage problem and not pre-zygotic stage problem like we saw before. Then another thing is that such mosaic women have high risk for development of cancer, gonadoblastoma. And now we have a question like, why are the females who suffer from Turner short-heighted? Okay, before we answer this question, we have to understand the Lyon hypothesis. So Mary Lyon was the scientist who formulated this hypothesis. And according to Mary Leon, she found her hypothesis of Leon hypothesis. She stated that females have two X chromosome, out of which they only use one X chromosome and keep the other X chromosome inactivated, and that inactivated X chromosome is called as bar body. And this happens that some cells are going to have, for example, in normal female, what happens according to this hypothesis, that maternal and paternal X chromosomes are coming, right, so that we get a female. So in some cells the maternal X chromosome will be active, and in some cells the paternal X chromosome will be active, and this happens totally by chance at the blastocyst stage of the development of zygote. That is, during very early stage of life, some cells will have paternal X chromosome active, while some cells will have maternal X chromosome active. So this is what is Leon hypothesis. And we should remember that the inactive X, that is the bar body, becomes a dense structure, that is, it is converted into heterochromatin and is hypermethylated. So if we see, normal females are also mosaic as far as inactivation of X is concerned. That is, some cells are going to have for paternal X active, and some cells are going to have maternal X active. So within the same female, there can be some two types of karyotypes found. Karyotype is basically same, but X paternal is active, X maternal is active. So that's why normal females are also mosaic, or they can be called as mosaic. So, according to Leon hypothesis, normal female just needs one X chromosome, and the other X chromosome is converted to bar body and is inactivated. So, what if female has extra chromosomes? Okay, even if the female has extra X chromosome, she activates only any one of the X chromosome and converts the rest chromosome into bar body. For example, if a female has only one X chromosome, she won't have any bar body. If she has two X chromosome, one bar body, that's female, normal female. If the female has three X chromosomes, she's going to have two bar bodies, and if she has four X chromosomes, she's going to have three bar bodies. According to some scientists, they say that extra X chromosome gives some poor mental developmental problems. And also in Turner females, we have noticed that Turner females, or females suffering from Turner syndrome, have spatial orientation problem. That is, they don't understand in corridors, like how far people are, how close they have to be someone, like they have problems in external environmental orientation. So, question is here that according to Leon hypothesis, one X is normal, one X is enough for the development of normal female, and the other is basically converted to bar body. But in Turner syndrome, why do we have Turner syndrome? Why? Because Turner syndrome has, classical system has 45 XO. So then why do people suffer from Turner syndrome? Only one X they have now. That one X is enough for their normal functioning. But no, they get Turner syndrome. So this means that the theory of Leon Mary failed to explain the development of Turner syndrome, even though she had just one X chromosome. So to solve this problem, the hypothesis was altered a bit by doing further studies. So after what we call further investigation and modification of the hypothesis, the new conclusion was that females selectively reactivate the second X chromosome, that was the bar body, during gametogenesis. And also, even in the bar body, some of the genes remain fully active. That means that bar body is not fully inactive, it is just partially inactive. So now we can say that the new Leon hypothesis basically will be like, one X chromosome in a female is fully active and the other X chromosome in a female is partially active. So according to new concept, the one X, either paternal or maternal, in some cells are going to be fully active, while the other is going to be partially active. And now coming back to our question, that why females in Turner syndrome are short heightened or short heighted. This is because height of female, the genes which are associated with the height of female are kept active on both the X chromosome. So in Turner syndrome female, they have only one X active and other is not present. In classical case 45X. So that is the reason why females who have Turner syndrome have short height. The gene which is associated with height is called as SHOX, that is short stature homeobox gene. It is present on the P arm of X chromosome. These SHOX genes remain active on both the X chromosome and help the female to achieve normal height. But in Turner, that is 45XO, one of the X chromosome is defective or absent, So the Turner female height is shortened. Now we are going to discuss about Down syndrome. So Down syndrome is the most common cause of mental retardation, and Down syndrome is also the most common chromosomal disorder. Down syndrome also we have classical case, special case and mosaic case. So in classical case of Down syndrome there is trisomy of 21st chromosome, and in special case there is translocation, example Robertsonian translocation, and in mosaic case this is mix. So what happens in classical case, there is actually trisomy of chromosome number 21 which causes Down syndrome. So basically the character will be 47, okay, because one extra chromosome, and XX, that is female, and XY male, okay. So the problem is there is an extra copy of X chromosome, extra copy of the 21st autosome, okay. So from where does this extra copy come? So this extra copy of 21st chromosome can come either from father or from the mother. That is due to a problem in the oogenesis, spermatogenesis, but in majority of case this extra 21st chromosome comes from the maternal side, okay. And the reason of this extra chromosome coming either from maternal or paternal side will be due to a prezygotic problem such as non-disjunction during first or second meiotic division. So if there was non-disjunction during first meiotic division, it will be due to the non-disjunction problem in the homologous chromosome pair, and if it was due to non-disjunction in second meiotic division, it was due to the problem that the sister chromatids didn't separate from each other. So this is the explanation for the classical case of Down syndrome, that is trisomy of chromosome 21. Okay, so the clinical features or special features of Down syndrome are that they have severe mental retardation, they have epicanthal fold with flat facial profile, so they will have folds near the eyes. Here we can see the epicanthal folds in the baby here. This is the epicanthal fold in a Down syndrome patient. And then they are basically good natured people and are not irritable. They are pleasant people. The patients who suffer from Down syndrome, they are also called as Prince Charming of the mentally retarded world. They have extra skin on the neck. It can be seen like this, the extra skin on the neck. And they suffer from congenital heart problems. They have umbilical hernia, gastrointestinal tract stenosis, hypotonia, and single horizontal crease. Because normal hands there are actually 2 to 3 crease in our hands, okay. But in patients who have Down syndrome, they just have a single crease, and that crease is called as simian crease. It is seen in the palm of the hands. So here we can see a normal palm which has 2 or 3 crease here also, whereas here there's just one crease, and this is a Down syndrome hand and this is called as simian line or simian crease. And the one more important feature is that they have low IQ and dribbling saliva. So just to recap, they have mental retardation, they have epicanthal folds with flat facial profile, okay. Their face is flat, they are good nature people, so they are foolishly smiling, and they have extra skin on the neck. They have congenital heart problems, they have umbilical hernia, GIT, stenosis, hypotonia, how we can see how their legs are relaxed, okay. And they have single hand crease, horizontal crease, which is called as simian crease, and they have low IQ and dripping saliva. That can be seen here, the dripping saliva. This is a patient who suffers from Down syndrome. Here I would like to mention an important information that previously most common cause of death of Down syndrome patient was severe infections at early age. But now due to the advancement of medicine, we have learned to control very severe infection. So due to this reason, the average age of Down syndrome patient has increased. But nowadays the new trend is that most of the Down syndrome patients die due to congenital cardiac problems. But if the patient is lucky, that is the Down syndrome patient is lucky and has no cardiac problems, then he or she may live for a longer period of time. But in the later stage of their life, they may suffer from Alzheimer's disease. That is, if the patient lives to around 40 years of age without any cardiac abnormalities, in his Down syndrome patient, he will eventually get Alzheimer's disease. And even if the patient survives somehow the Alzheimer's disease, then they have a high risk of developing acute leukemia and eventually die. So now scientists are studying the link between genes on chromosome 21 with Alzheimer's disease and acute leukemia. Like, what is the relation between these diseases and the genes on chromosome 21? The chromosome 21 has around 225 genes. And one more thing to remember is that patients who are mosaic, they have a high risk to develop gonadoblastoma, which is a post-zygotic problem that patients who are mosaic. Mosaic is due to post-zygotic problem. So in mosaic, basically, both the father and the mother, the paternal and the maternal lines gave equal number of chromosome, proper number of chromosome, but during the early stages of the fetal life, there was some problem with what we call mutation, and some lines of cells started to have extra 21st chromosome, and that led to mosaic situation, and then such people have high risk of developing gonadoblastoma. The most important point associated with Down syndrome is that it is strongly associated with increasing maternal age. So increase in maternal age is directly proportional, maternal age is directly proportional to Down syndrome. So as the maternal age increases, the risk of Down syndrome also increases. And according to statistics, 45 years of age women who give birth will have one down in every 25th baby. So after the age of 45 years old, women gives birth, there's a risk of every 25th baby being a Down syndrome patient. So we discussed the classical case of Down syndrome, that is chromosome 21 trisomy, and we discussed the general features of Down syndrome patient. Now we will see the special case of Down syndrome. So 4 to 5% of Down syndrome cases are due to translocation. That is what happens. For example, if this is chromosome number 14 and this is chromosome number 21, both these chromosomes will break near the centromere, okay, and both of these chromosomes, only their long arms will fuse with each other. So this means their lost short arms will be lost and their long arms will fuse as such. And this kind of translocation is called as Robertsonian translocation. So Robertsonian translocation is where two acrocentric chromosomes have lost their short arms and their long arms are fused together to form a new chromosome, and this is called as Robertsonian translocation. So just as a quick recap, metacentric have equal centromeres, okay, they have equal short arm and q arm. Submetacentric have short p arms and long q arms, whereas acrocentric have very short p arms and very long q arms. So in Robertsonian, what we call translocation, the very short p arms are lost of both the 14th chromosome and the 21st chromosome. And then the long arms fuse together, it is 14q and 21q, and result in Down syndrome. Note, if Down syndrome patient suffers or inherits a extra dose of 21 due to Robertsonian translocation, because this is an extra dose of 21, right, then this will be a familial case of Down syndrome. We have to remember that about 95% of cases of Down syndrome are sporadic and not familial. Only 4 to 5% cases of Down syndrome are familial, like in Robertsonian translocation. So if most of the cases of Down syndrome are not familial, in family, if one of the child suffers from Down syndrome, there is no increased risk to have another Down syndrome kid, unless on only exception is that if the maternal age is high, the risk of having Down syndrome kid is high. Now our question is why maternal age is associated with Down syndrome or other inherited cytogenic disorders. So we should remember that female fetus, even before the fetus is born, or it's the fetus itself is in the fetal stage, within the ovaries of the fetus, the first meiotic division starts and the ova are arrested at diplotin stage of the first meiotic division. After that, the female keeps all those cells arrested in the diplotin stage. So in diplotin stage, what happens that the chromosomes and genetic material are loosely arranged and are very vulnerable to environmental and mutagenic influences. So when the female is born and she reaches the age of puberty, then menstrual cycle starts, and every month She reactivates some of these cells from the diplotin stage and completes first and second meiotic division, and so the ova is formed. Now, as the female age keeps on increasing, her premature gametes with open genetic material which are arrested in diplotin stage have more and more chance to get influenced by mutagenic and environmental factors. As a result, it can lead to different problems, pre-zygotic problems in oogenesis, such as non-disjunction during first meiotic division, non-disjunction during second meiotic division, and even anaphase lag. Nowadays, females who are older than old age who get pregnant, or female who are older than 40 years of age, when they get pregnant, they go for prenatal diagnosis to rule out whether the baby has Down or not. So prenatal diagnosis is done by taking amniocentesis or chorionic villus biopsy, and then they do the karyotypic studies. Other simple tests which help to estimate the risk of Down in old pregnant females is that it's called a triple screen test. So in this, what we do, that we check the level of alpha fetoprotein, HCG, and conjugated estriol in the blood of pregnant female. If the values are out of normal range, 70% of Down syndrome can be diagnosed with this test. Okay. But to confirm the diagnosis of Down syndrome, we need to do amniocentesis or chorionic villus biopsy to get the genetic material of the baby and do karyotypic studies to look for chromosome 21 trisomy, and then we can confirm whether the baby has Down syndrome or not. The risk of having Down syndrome is pretty high, and the risk of Down syndrome is 1 in 700 pregnancies. That is the statistically proven. So the risk of Down syndrome is very high. Now we are going to discuss another syndrome, that is the Edwards syndrome. So Edwards syndrome is caused due to trisomy of chromosome number 18. The prevalence of Edwards syndrome is 1 in 8,000 cases of 8,000 cases of pregnancy, so it is much lower than as compared to Down syndrome. So the karyotype of Edwards syndrome is going to be 47XX. So the extra chromosome is the extra 18th chromosome, and the reason can be pre-zygotic. That can be due to non-disjunction during the first or non-disjunction during the second meiotic division. So in Edwards syndrome, it can be problem either with oogenesis or spermatogenesis. That is in the pre-zygotic stage. That is non-disjunction can be in either first or second meiotic division. Important thing to remember: Edwards syndrome can also occur in both males and females, but it is more prevalent in females. Okay, the symptoms and signs of a person who suffers with Edwards syndrome are: so they have a very prominent occipital bone, and the chin is pushed inward. So it is somewhat like this, that the occiput is very big and the chin is inward. It is like someone punched from here and the occiput went out from that side. So that is one of the signs of Edwards syndrome. Then secondly, this inward pushing of chin is also called as micrognathia. And then their ears are lower as compared, because normally the level of eyes and ears is same if you see from side of the head, but here the ears are lower as compared to the level of the eyes. And these patients are mentally retarded, they have a short neck, they have congenital heart disease, which is common in even in Down syndrome, in Edwards syndrome, and as well as Patau syndrome. And then they have renal malformation. The abduction of their leg is limited, that is legs are close to their body, whereas we remember Down syndrome the patient had hypotonia, so his legs were spread. And the special feature is that the sole of the foot is curved, and that is called as rocker bottom feet, and they have overlapping fingers. So here we can see an example of a diagram of Edwards syndrome. So the patient is mentally retarded, very prominent occiput bone. The chin is inward, called micrognathia. The ears are lower as compared to the eyes. There is very short neck. The patient suffers from congenital heart disease. The overlapping fingers of the hand are overlapping. There is renal malformation, abduction of leg, that is legs is very close, they can't open the legs much, and they have rocker bottom field. So this is Edwards syndrome. Now if we compare Down and Edward, what are the similarities? So both Down and Edwards syndrome, the patients are going to be mentally retarded. Both of the patients suffering with Down syndrome and Edwards syndrome are going to have congenital heart defects. The differences between Down syndrome and Edwards syndrome are that in Down syndrome epicanthal folds are present. In what we call Edwards syndrome, epicanthal folds are not there. In Down syndrome, what we call the face is flat. It looks like someone has punched from here. But in Edwards syndrome, it looks like someone has punched from the chin and the head goes out. The face is not flat. In Down syndrome, the patient has flat normal feet, whereas in Edward syndrome, he has rocker bottom feet. That is because his feet is curved. That is why it is called as rocker bottom feet in Edward syndrome. In Down syndrome, the patient has hypotonia, so his legs can be widespread, whereas in Edward syndrome, the patient has very low abduction in the legs. So this was some similarities and differences between Down syndrome and Edward syndrome. Now we will discuss Patau syndrome. So Patau syndrome is caused due to a trisomy of chromosome number 13. The signs and symptoms of Patau syndrome are the patient is going to have microcephaly, that means he has a very small head, and has microophthalmia, that means has very small eyes. The patient is mentally retarded. He has heart defects. He has renal problems. He has hernia, that is umbilical hernia, and rocker bottom feet and cleft lips. This is the example what is cleft lip. So now if we compare all the three problems, that is Patau syndrome, Down syndrome, Edwards syndrome, all three are going to be mentally retarded patients. All three are going to have congenital heart defects. Okay, the differences are that a mentally retarded patient who has microcephaly and microophthalmia is Patau syndrome. A mentally retarded patient who has a flat face with epicanthal folds is Down syndrome, and a mentally retarded patient with whose chin is inward and has prominent occiput bone, we are going to think that it is going to be Edwards syndrome. Then some other problems are that in Patau we have renal problems as well as in Edward, but in Down we don't have renal problems mostly. We have umbilical hernia in Patau, we have umbilical hernia down. In Edward we don't. Now we are going to discuss the structural defects in chromosome. So the one of the most important structural defect in chromosome is translocation. So translocation means that is genetic material is broken from one chromosome and it is transferred to another chromosome. Okay, so basically there is shifting of genetic material from one side of the one chromosome to another chromosome. For example, we should know that translocation is of three types. There is simple translocation, reciprocal translocation, and Robertsonian translocation. What is simple translocation? Simple translocation means there is transfer of genetic material just from one chromosome to another. Nothing is given back. So for example, chromosome 9 will transfer some of its genetic material to chromosome 29 and doesn't get anything in return. So this is simple translocation. The next type of translocation is called as reciprocal translocation. So to explain that, here I have an example. For example, this is a blue chromosome, this is a red chromosome. So both of them are going to exchange material. Okay, so for example, the blue cap is going to come on the red pen and the red cap is going to come on the blue pen, and so we will have this such result. So this type of translocation is called as reciprocal since it is two-way. The total number of chromosome remains normal in reciprocal translation. The person does not suffer because total genetic material is still the same, that is no genetic material is lost, but this person is going to make gametes, and during gametogenesis the problem may be present. Okay, in when there is reciprocal translocation, sometime reciprocal translocation may become problematic even for the person who has the translocation. So it is quite possible that the reciprocal translocation might not cause any problem to the person who has it, but when he forms gamete there might be a problem. Or it can be like even when he forms gamete there is a problem, and even for the person the reciprocal translocation will cause problem. So what are the example of such problems caused by reciprocal translocation? So one such example of that reciprocal translocation is in B lymphocytes. So B lymphocytes is chromosome number 4, has chromosome number 14. On the chromosome number 14 of B lymphocytes, there is a gene for immunoglobulin gene which is very hyperactive. Okay, so sometimes what happens, we have two other genes. Okay, so for example, if we count chromosome number 8, this part of chromosome number 8 which has the MYC gene, if it translocates, reciprocal translocation occurs and MYC gene comes to the chromosome 14, and this lower part of chromosome 14 goes to chromosome number 8. So now this chromosome number 14 is going to have immunoglobulin G G, immunoglobulin G gene as well as MYC gene. So we already know the immunoglobulin G gene is very active, and MYC gene in the presence of immunoglobulin G hyperactivity will also be expressed in higher rate and overexpressed, and as a result MYC gene is responsible for proliferation of cell, and as a result the B lymphocytes are going to proliferate so much, and that will lead to Burkitt's lymphoma. Another possibility with B lymphocytes of reciprocal translocation is that if the chromosome number 14 translocates with chromosome number 18, so chromosome number 18 bottom up we have BCL2 gene. Okay, if this BCL2 gene will translocate and come on this part of chromosome 14, and this part of chromosome number 14 goes there, so BCL2 gene will be present in the vicinity of hyperactive immunoglobulin G gene, and then as a result, due to high activity immunoglobulin G gene, BCL2 gene also starts acting at a higher rate, and that causes the elongation of lifespan of beta lymphocytes. Okay, BCL2 gene is responsible for lifespan, whereas MYC gene is responsible for proliferation. So BCL2 gene, when it is present in the vicinity of IgG on chromosome number 14 after reciprocal translocation, will overexpress and cause the cell to live for a longer period of life, leading to follicular lymphoma. So follicular lymphoma. Another example of reciprocal translocation is when reciprocal translocation associated with hematopoietic cells in the bone marrow. Okay, here what happens that in hematopoietic cell, chromosome number 2 has BCR gene and chromosome number 9 has ABL gene. A large part of chromosome number 2, chromosome number 22 shifts to chromosome number 9, and a very small part, the lower part of chromosome number 9, basically shifts to chromosome number 22. This means that the ABL gene and the BCR gene, okay, they will fuse on they will be present on the same chromosome now, and they will fuse together. Okay, and BCR gene is basically called as breakpoint cluster gene. The fusion of BCR and ABL gene is decent, and they make mRNA. Okay, so the result of this gene formation will make an mRNA, and that mRNA will have some complementary area of BCR and some complementary area of ABL, and this mRNA will be translated into a pathological protein. So this mRNA will be translated into a pathological protein, and that protein is called as chimeric protein. And this protein, when it expresses on the surface of hematopoietic cell, it will give strong signal for overproliferation, and as a result it leads to cancer, especially chronic myeloid leukemia. Another important point to remember is that this was an example of also what we call reciprocal translocation. Apart from that, the chromosome that lost a large part to chromosome number 9 and got a small part from chromosome number 9, that hybrid chromosome is called as BCL-ABL hybridization or Philadelphia chromosome. So this chromosome was basically Philadelphia chromosome, which is formed after the exchange of reciprocal translocation that led to the formation of abnormal mRNA, that led to the formation of pathological protein called chimeric protein that leads to mostly chronic myeloid leukemia. So now we have discussed simple translocation and reciprocal translocation. Now we are going to discuss another type of translocation, that is Robertsonian translocation. So Robertsonian translocation is seen in between chromosome number 13, 14, 15 and 21. There is something special about these chromosomes. These chromosomes are mostly acrocentric chromosomes. So what are acrocentric chromosomes? So we have types of chromosomes. So a metacentric chromosome is a chromosome in which the centromere is in the middle, whereas in submetacentric the chromosome is slightly away from the middle, and in acrocentric chromosome the centromere is towards the end of the chromosome. So the one part is very long, one is very short arm. And Robertsonian translocation is seen mainly in acrocentric type of chromosomes, and 13, 14, 15 and 21 chromosomes are all acrocentric chromosomes. So what basically happens in Robertsonian translocation is two acrocentric chromosomes, okay, irrespective, okay, no random, any of these two acrocentric chromosomes, they will fuse together and form a chromosome which has only long arms and other chromosome which has only short arms. So for example, here this is chromosome number 21 and this is chromosome number 14. So it has long and short arm, long and short arm. So when these two chromosomes are going to fuse together, they will form a chromosome which will have the 21st chromosome's long arm and the 14th chromosome's long arm, whereas they will form another chromosome which is going to have only the short arms of both the 14th and 21 chromosome respectively. The small arm complex usually doesn't get centromere, okay, and so it's destroyed and gets digested away. Now, after the translocation, the total number of chromosomes in the cell decreases to 45. Why is this so? So here, as we can see, we had two chromosomes. For example, if this is the head and this is the body, this is the head and this is the body, we threw both the heads and we just kept the body. So eventually we will have one person. We won't have two persons anymore. This person is no more, right? So this means the same if you apply for chromosome. We had two chromosomes, but in the end we have just one remaining chromosome because this is thrown away or destroyed. So due to this reason, the total number of chromosome is now not 46, but it is 45. The total number of chromosomes is reduced. So to recap, Robertsonian translocation is fusion of long arms of acrocentric chromosomes and the short arms are lost or destroyed. Now the question is, even if in this particular cell or in this particular person the number of chromosomes has been reduced to 45, the person or the cell doesn't suffer from any problems. Why is that so? This is because the person who has Robertsonian translocation or cell which has Robertsonian translocation doesn't suffer because we know only the short arms of the acrocentric chromosomes will be lost, right? And because short arms of acrocentric chromosome have genes for formation of ribosomal RNA. So if some short arms of some acrocentric chromosomes are lost, there are multiple other acrocentric chromosomes along with the short arms which are totally intact. So these other normal acrocentric chromosomes which have no Robertsonian translocation, they will have the same genes on their short arms, and those genes can contribute to the formation of the ribosomal RNA. And as a result, the affected cell or the affected person doesn't suffer any consequences even after loss of few short arms of few of his acrocentric chromosomes. And also to understand it better, in acrocentric chromosomes the important genes are present on the long arm, and so in Robertsonian patients have all long arms of all the acrocentric chromosomes, so they won't suffer because the set of important genes which they need is complete because they have the long arms. The only thing they lost is the short arms of some acrocentric chromosomes which result in the formation of Robertsonian translocation. This is the reason why Robertsonian translocation doesn't make a problem for the patient or the cell affected by it. Okay, now to understand, 1 in 1000 people are Robertsonian people. The problem is that Robertsonian translocation will cause problem only when the person, he or she, is going to make gametes, because Robertsonian chromosomes have trouble that they should exchange material with 14th or 21st chromosome. So what is this? We are going to understand in more detail, but before that we need to remember that Robertsonian translocation patient produce abnormal gametes and there is repeated loss of product of conception. This also we are going to see now why so. And male or female, anyone can be Robertsonian. So now our question is, how abnormal gamete formation is going to make trouble for the Robertsonian patient. So for this we are going to consider a healthy wife and a Robertsonian husband. Okay, so we know that the woman will undergo oogenesis, okay, and she will form the gamete side. So we will consider here chromosome number 14 and 21. So after the oogenesis, after meiosis 1 and meiosis 2, the gametes formed by the female are each going to have one copy of 14th and 21st chromosome, okay, that we know. But in the husband, the possible what we call scenario of the sperms we will see as of now, because he has one 14th chromosome, one 21 chromosome, and one chromosome which has what we call as 14q and 21q. So what are the possible sperms he can produce? So if this person will combine the first two and leave this in the other gamete, right? So we will have this kind of sperm. So if this sperm fuses with a normal egg, we are going to get two 14 chromosomes and 121 chromosome, and what we call another 21 chromosome, but there's also 14q. So this means that there is trisomy of 14, and we remember that trisomy of 14 is not compatible with life. So due to this reason, the baby will be aborted. Now if we combine this gamete with the normal gamete of the wife, we are going to get monosomy of 14, and we remember monosomy of autosomes is also not compatible with life. Due to this reason also, the baby will be aborted. The second possibility that the male can synthesize sperms are that he will combine these two he will combine these two as one, and this goes separately. So when we fertilize such sperm with normal female egg, we are going to get trisomy of 21. As we can see, 21, 21 and 21q, that means there is trisomy of 21 chromosome, and we know trisomy of 21 chromosome causes Down syndrome. So the baby who is born is going to have Down syndrome. And if we take this sperm and fuse it with the normal egg, we are going to get monosomy of chromosome number 21, because we just have 121 chromosome, and we know that monosomy of autosomes is not compatible with life. So this will also not be compatible with life, and the baby will be aborted. Now if the couple is persistent and they try again, the possibilities is that maybe the husband makes a sperm which has only the Robertsonian chromosome separately, and the other chromosomes go to the other sperm. So if this sperm fuses with the normal egg, we are going to have such possibility that we have 14 chromosome, 121st chromosome, and we are going to have 21q and 14q. So this means that the baby is going to be normal, okay, but he is going to be a Robertsonian carrier. So he himself won't have any problem, but when in future he will also form gametes, he will have a lot of problem forming the gametes, and there can be highly risk of most of babies getting aborted. Then finally, the last case, when the husband has absolutely normal gametes 14 and 21, and the female also had 14 and 21, the baby is going to be healthy, normal, with very good karyotype. So this is the reason why Robertsonian translocation will cause problems with gamete formation, and why there is repeated loss of product of conception. So in this example, we saw out of 6 kids, 3 were aborted, 1 kid was Down syndrome, 1 kid was normal, and 1 kid was normal but he had Robertsonian translocation and acted as a carrier, and that Robertsonian translocation kid who acted as a carrier has only 45 chromosomes.